So I just wanted to go ahead and welcome everybody today. It is just awesome to have you here today. And I always look forward to Symposium because I always learn something from all of the interns. Just want to say thank you to the interns for their hard work, and thank you to all of our mentors who could join us today. We just know that you have done an awesome job in encouraging and supporting and teaching and mentoring our students in the program. At the beginning, there was a little bit of a reminder that if you do have questions of the interns, wait until their presentation is finished to put those into the chat so that you don't distract them during their presentation. Each intern is going to have 10 minutes to present, and then we just have a couple of minutes for questions. And once again, it is just so great to be able to hear these presentations. And again, thank you to the interns, to the mentors, to co-mentors, to everybody. It's going to be a fantastic presentation time. So with that, we are going to go ahead and get started with our first presentation. This is going to be a team presentation. And this is Amelia and Keya. They are going to be talking about, now, is it tracking Trk-B, or do I say that differently?
I believe it's pronounced tracking Trk-B. We kind of wanted to do a play on words there.
Okay, thank you. And they're going to be talking about a very neat idea that they came up with the follow-up Parkinson's disease study proposal. So look forward to hearing from them. And I'm going to go ahead and turn off my video and mute myself. So please go ahead. Thank you.
Thank you, Linda. So I'm Keya Pandya from Jesuit High School.
And my name is Amelia Kotamarti from Grant High School.
And this is our presentation on tracking Trk-B, a follow-up Parkinson's disease study proposal.
Yes, and our wonderful mentor is Dr. Charles Meshul, and we both interned this summer at the VA Portland Healthcare Systems. Alrighty, so our internship this summer was a little bit unorthodox because we weren't able to physically be in person in the lab. But I just kind of wanted to give you guys an idea of what our internship actually looked like. So it was kind of broken into three parts. We first were able to really dive deep into the details of Parkinson's disease, and in that we were able to do this via lectures and discussions. In our second part, we actually were able to look at some of the current research that's being done, so we critiqued and read journal articles, including one of Dr. Meshul's. And then in this last little bit, we've been preparing our presentation for you today.
Yeah, so like Amelia said, we weren't in person. So of course we were a little bit disappointed that we didn't get to perform those experiments or lab procedures that ordinarily interns would be able to. But there was definitely a huge silver lining this summer, which was that we definitely learned a lot more detailed kind of technical information and a lot more research skills.
Yeah. Alrighty, so our presentation today is going to kind of be broken into three parts. First, we're going to give you guys an overview of Parkinson's disease itself. Then we're going to discuss a study that was actually published by our mentor, Dr. Meshul, and a few of his colleagues back in 2020. And then we are going to propose for you today a follow-up study design.
Yep. So just kind of introducing Parkinson's disease and a little bit of neuroanatomy that you're going to need to understand the rest of our talk. Parkinson's is a progressive neurodegenerative disease, which is caused by a lack of the neurotransmitter dopamine in the midbrain. And that deficit is caused by dopamine loss in two parts of the brain called the substantia nigra and the striatum. So Parkinson's, like the majority of other neurodegenerative diseases like ALS and Alzheimer's disease, is not really able to be diagnosed until the symptoms, and in this case, motor symptoms, present themselves in a patient. And the issue is that the disease starts about 25 years before these motor deficits can even be observed in patients. So kind of by that point, once the disease is diagnosed, it's already too late to start any preventative treatments. So the treatment is then kind of focused on mitigating symptoms and improving the quality of life for patients. So again, while there are long-term kind of treatments for symptomatic relief, there are not currently any treatments that would stop or slow the progression of the disease once it is started. So just introducing a little bit more about neuroanatomy. So the substantia nigra, the part of the brain that I just brought up, contains our key dopamine cells. And the substantia nigra then projects to or communicates with the striatum, which contains dopamine synapses. And both of those parts of the brain communicate with the motor cortex through kind of a complex brain circuitry. Yeah, so just introducing a little bit about dopamine synthesis quickly. Dopamine is synthesized in synapses, as you can kind of see on that figure in the slide there. So dopamine synthesis begins with an amino acid called tyrosine. And the enzyme tyrosine hydroxylase is found in dopamine cells in both the striatum and the substantia nigra. And it turns that tyrosine into L-DOPA. There's then a second enzyme called dopa decarboxylase that then turns L-DOPA into dopamine. And you can kind of see that on that little pathway that we've created on the right there. So there are a couple of reasons why it's important to understand dopamine synthesis. One is because the intermediate between tyrosine and dopamine is actually involved in one of the most common forms of treatment for Parkinson's. So this treatment is called levodopa therapy, and essentially it replaces the lost dopamine in the brain. So the L-DOPA enters the brain and is then metabolized into dopamine in the synapses. And like any treatment, there are a few negatives. The effect of L-DOPA does decrease as the disease progresses because, again, it's only symptomatic relief and it doesn't really stop or slow that progression of the disease at all. Diving into kind of another reason why it's important to understand dopamine synthesis is because of the importance of tyrosine hydroxylase in today's talk, as well as in Parkinson's pathology. So tyrosine hydroxylase is actually one of the primary ways that scientists are able to label and mark dopamine cells. So kind of wherever there is tyrosine hydroxylase, there's also dopamine. So it's kind of a very reliable indicator as to where dopamine has been depleted, what cells have died, and how much degeneration has kind of occurred overall.
Yeah, and so now I'm going to go over the Trk-B receptor signaling cascade. So Trk-B is a receptor naturally found in the brain, and it is stimulated by a chemical called BDNF. So BDNF stands for brain-derived neurotrophic factor, and it is a growth factor that, as the name suggests, promotes brain plasticity and survival and growth. So the Trk-B receptor is actually a specific type of enzyme called a kinase. So kinases are specialized enzymes that can activate other enzymes, and they do this via a process called phosphorylation, which is very important to our talk. So phosphorylation is essentially where a kinase will add a phosphate group onto another enzyme and kind of transfer it into its active state. So for today's purposes, phosphorylation equals activation. So what's neat about this Trk-B receptor is that it phosphorylates or activates other kinases. So then these kinases are able to activate yet other kinases, and it just starts this whole domino effect, which you can kind of see in the figure to our right, of all these kinases activating one another. So why is the Trk-B receptor signaling cascade really important to Parkinson's disease researchers? Well, Parkinson's disease is characterized by degeneration and cell death, and the Trk-B receptor, when it's stimulated by BDNF, promotes survival and brain plasticity and growth. So there is a hope that the mechanism behind Trk-B can somehow be involved in a way to intervene with the degeneration seen in Parkinson's disease.
So just moving into kind of introducing a couple mouse models that we're going to be talking about both in our study proposal and in the study from the Meshul lab that we're going to go over a little later in the talk. So the first model is the MPTP mouse model, which is used in Dr. Meshul's study. So the MPTP model is a toxin-based model where MPTP enters the brain and is then metabolized by an enzyme and is taken up by dopamine transporters, which is kind of why this model only attacks the dopamine-producing neurons in the brain and not really anything else. So that MPTP is metabolized into a neurotoxin called MPP+. That MPP+ then kills dopamine-producing neurons in the substantia nigra, and it kills these neurons by kind of mucking up the mitochondria that are present in the neurons, which leads to kind of this buildup of toxic molecules that then contributes to cell death and cell destruction. So in the context of Dr. Meshul's study, the lab utilized a progressive model of MPTP, which was delivered over the course of 4 weeks to model the progressive and degenerative nature of Parkinson's. And then moving forward to the mutated alpha-synuclein model, just as a little bit of background information, one hallmark of Parkinson's pathology are something called Lewy bodies, or these aggregated clumps of protein that are found in the brains of patients. So the primary protein that's aggregated in these Lewy bodies is called alpha-synuclein, which makes it kind of a protein of interest in the underlying cause of Parkinson's. So normally, alpha-synuclein is a protein that regulates synaptic vesicle trafficking and neurotransmitter release. And with this model, basically, it's a chronic overexpression of the mutated form of alpha-synuclein via something called the adeno-associated viral vector system, or the AAV vector system. So the mutated form of alpha-synuclein basically just makes it a little bit more toxic and more susceptible to that aggregation. And just kind of for future reference, the AAV vector system takes advantage of the capability of viruses to easily enter cells, and it uses them as these little transport vessels for whatever protein coding gene scientists want to introduce into cells. So the introduction of that mutated version of alpha-synuclein causes aggregation of the alpha-synuclein, which causes dopamine cell death.
Alrighty, so now we're going to transition into the second part of our talk. So we're going to kind of cover a paper that was published back in 2020 by our mentor, Dr. Meshul, and a few of his colleagues. And so it's looking at the effects of a prospective treatment in this progressive NPTP mouse model that Keya just described.
Yeah, so just kind of as a refresher on some of the key terms that you're going to need to understand Dr. Meshul's paper and our study proposal. Starting off with Trk-B, Trk-B again is just a receptor that's found in the brain that is stimulated by BDNF, or brain-derived neurotrophic factor. So by exciting that Trk-B receptor, BDNF also stimulates that kinase domino effect that Amelia went over earlier that promotes plasticity and brain growth. So DHF, this is a new one, or 7,8-dihydroxyflavin, is the drug treatment that was tested in Dr. Meshul's study. So the DHF stimulates this Trk-B receptor. So, essentially it's a replacement for BDNF that's just a little bit more clinically viable. So this made DHF look pretty promising as an alternative treatment for Parkinson's.
Yeah, so now I'm just going to go over a little bit about antibodies and staining. So in this experiment and in many other experiments, scientists need to be able to accurately visualize those invisible microscopic changes that occur in the tissue of their models as a result of whatever treatment they're testing. And so they can do this in many different ways, but one of the primary ways many scientists do this is through a process called immunostaining. So as many of you know, antibodies are used in the body to latch onto and label foreign molecules to kind of trigger a protective immune response. And so scientists are able to actually use this to their advantage. So what they do is they engineer these specific antibodies that will only latch onto and label their proteins or chemical species of interest. And so what they're able to do is they're able to make these antibodies colorful or fluorescent or visible in another way. And then they apply their antibodies to their tissue and sample, and it'll only latch onto and label exactly what they're looking for. So there are plenty of ways to apply immunostaining, but the two primary ways that we're going to be talking about today are immunohistochemistry and Western blots. So both of these tests are semi-quantitative, but this right here is immunohistochemistry. And as you can see, that antibody stain was kind of applied to an intact piece of tissue. So you get a very visual and anatomically specific stain that can tell you a lot about where the specific proteins of interest are. And over here, this is Western blot analysis. So this is when the antibodies are applied to broken down tissue, and therefore there's no real anatomical specificity, there's no visual component to it. But the thing is it's more quantitatively accurate than immunohistochemistry, so it's often favored for quantitative analysis. And now just launching into the results of this paper, if you'll remember, this is studying that BDNF replacement, the DHF treatment, in our progressive NPTP Parkinson's model. So we were able to see some really encouraging benefits as a result of that DHF treatment. And just as a caveat, we are going to be discussing kind of the most relevant results to the topic of our talk, to the subject material that we're going to be covering, and also what we thought were kind of the most consequential results of this paper. So one of the really important benefits that we saw was gait improvement. So gait was measured by something called the DigiGait apparatus, which you can see right up here. So the DigiGait apparatus is essentially able to measure a bunch of different aspects of a mouse's movement while they run on a treadmill. And so an example of one of these measurements is called swing time, which I have right here. So as you can see, so swing time is basically an indicator of motor deterioration because the longer the swing time of the paws, the more motor deterioration there is. And so right here you can see in our control, there's a relatively low swing time. and it increases with our toxin model. And then right over here, it actually goes back down to below the control level when we add our DHF treatment, which indicates a pretty significant gait improvement. Another great result that we saw was we found that there was tyrosine hydroxylase recovery in the striatum. So if you'll remember, the striatum is that part of the brain that has those really important dopamine synaptic contacts, and tyrosine hydroxylase is the enzyme that's involved in dopamine synthesis. So the significance of this, which you can kind of see are stained down here, is that getting that tyrosine hydroxylase recovery in the striatum indicates that there is an increase in dopamine synthesis occurring in the striatum as a result of our treatment. And then it also further signifies that we are seeing a recovery of these synapses, which is very important. And another great benefit that we saw was an increase in our Trk-B signaling cascade activation. Activation can be kind of measured by phosphorylation, because if you'll remember, phosphorylation equals activation for today. And so the example kinase that I'm going to be talking about is AKT. So this is just one of the enzymes involved in that whole cascade we were talking about. So as you can see in this graph right here, with our toxin treatment, the amount of phosphorylation or activation of this kinase went down, and then it was recovered with our DHF treatment back to control level. So this kind of indicates that we're seeing, as a result of this DHF treatment, a recovery in that cascade activation and an increase in brain plasticity.
Yeah, so just kind of moving into a few of the deficits or holes that we saw in the results of this paper. So like Amelia said, there was tyrosine hydroxylase recovery in the striatum, but unfortunately there was not any recovery in the substantia nigra. So this means that even though there was an increase in those dopamine synapses, there was no really indicated cellular regeneration there. And you can kind of see that where the figure on the right is kind of data from a Western blot of midbrain tyrosine hydroxylase protein expression. And then the figure on the left is the tyrosine hydroxylase immunostaining. And from that immunostaining, you can see in that vehicle or control, there was kind of a lot of tyrosine hydroxylase. And then as we move to the MPTP treatment, there was kind of a very observable decrease. And then with that DHF treatment, there wasn't really any significant recovery there. So moving on, there was also no statistically significant dopamine tissue recovery. And that being said, there was a slight increase in striatal dopamine, which could be clinically significant since there was an observable alleviation of those motor symptoms in the mice. So it's just kind of important to make that distinction between clinically significant and statistically significant, because the question there then is how much kind of dopamine tissue recovery do you really need to kind of gain back those motor symptoms? So clearly, kind of according to these results, DHF does appear pretty promising as a potential prospective treatment for Parkinson's. Now, of course, there are still a lot of unknowns with this DHF treatment, and keeping that in mind, we've created a few follow-up study proposal ideas that would address a few of these unknowns.
Yeah. So as we're transitioning into the last little bit of our talk, I just wanted to do a quick review on our two primary models. So the MPTP model is the one that was used in the DHF study, and it's our progressive toxin that is administered over 4 weeks. And if you'll remember, that MPTP is metabolized into the neurotoxin MPP+, and then this is what eventually kills dopamine cells in the substantia nigra. And then over here with our alpha-synuclein model, this causes dopamine cell death in the substantia nigra as well, but through a different mechanism. So again, we use that viral system to inject and introduce mutated alpha-synuclein directly into the substantia nigra. And then the mutated version triggers this protein aggregation, which you can kind of see right here. And that is what causes dopamine cell death in the substantia nigra. So the reason why we're also really emphasizing this alpha-synuclein model is because we actually found a really intriguing paper that we read about how alpha-synuclein, the protein, chemically interacts with our Trk-B receptor. So the paper said that Trk-B is actually inhibited by this alpha-synuclein interaction. And so we thought it would just be really neat to look at this model because we think it would be interesting to investigate the interaction between this Trk-B receptor and our alpha-synuclein protein in the context of an actual in vivo Parkinson's disease model. So now transitioning into our study proposals. So we were not able to be in person in a lab and do experiments, so we kind of wanted to do a little hypothetical. So these are some of the things that we would do if we had space in a lab and if we had potentially unlimited funding and time and resources. So one of the first things we would love to do is to test that DHF treatment in our alpha-synuclein model for the reasons I just outlined. So we just think it's really neat to investigate that Trk-B alpha-synuclein relationship in an actual in vivo model.
Yep. And then moving into our second idea, which was kind of administering that DHF treatment along with levodopa therapy in both the MPTP and alpha-synuclein models. So if you'll remember, levodopa replaces that lost dopamine in the brain, and then DHF mitigated those motor symptoms through kind of a separate mechanism. So we thought it would be really interesting to see a treatment that would utilize both.
Yeah, and then we also thought it would be neat to see what happens if we take Trk-B out of the equation and see how this affects Parkinson's disease pathology. So this could be done in either of the models, potentially. And we also think we could do this pharmacologically by blocking the effects of Trk-B using drug treatment, or it could be done genetically, where you just knock out the Trk-B receptor in its entirety and see how that affects Parkinson's pathology.
Yep. So moving into our last idea, which is the overexpression of Trk-B. This is the idea that we kind of settled on and ran with and developed into a little bit more detail. So I will transition into explaining that a little bit now. Yeah, so our plan is to overexpress Trk-B through a viral vector system in 2 different mouse models. So it's the same 2 models, the MPTP toxin model and then the alpha-synuclein, using that AAV viral vector model that I went over earlier. And we'll go into that in a little bit more detail in a bit. But just to clear things up really quickly, when we say overexpression, we just kind of mean that we would make it so that the gene that codes for Trk-B would be just a little bit more active.
Yeah. So now just going into the why of our study proposal, why we chose this specific one to expand on. So the DHF treatment showed a lot of promise as a prospective treatment for Parkinson's disease, and its intent was to work by activating that Trk-B receptor signaling cascade. But because it is a pharmacological drug treatment, there could be side effects that we don't know about. DHF could be causing some of these effects or results via a different mechanism. And so what we really want to do is eliminate any doubt that's caused by it being a drug treatment and just focus on the role of that Trk-B receptor in our Parkinson's disease pathology. So that's why we're doing genetic overexpression as opposed to a pharmacological drug treatment. We also thought it would be neat to do that MPTP model specifically because the DHF treatment used it, and so our results would be a little bit more easily comparable with theirs. And then we wanted to use our alpha-synuclein model because of that really neat interaction between the alpha-synuclein protein and the Trk-B receptor that was described by that paper. And then we also think it would be neat to investigate how the efficacy of this proposed treatment would vary between those models, and we think that could reveal something about Parkinson's pathology.
So just now introducing kind of our proposed timeline for this study. Of course, first we want to start those Parkinson's model treatments. So we would start one group of mice with a localized injection in the substantia nigra of the mutated form of alpha-synuclein via that AAV viral vector system. And then we would start the other group with intraperitoneal injections of MPTP. So the treatment would then occur about 4 weeks later, where we would administer localized injections again in the substantia nigra of AAV delivered Trk-B, or the human Trk-B receptor via that viral vector system. And then kind of moving into kind of data collection and analysis, we do want to perform that same gait assessment using that DigiGate apparatus that you can see a picture of again up there. And we'd want to perform histological or tissue analysis. So that would kind of include Western blots, which we want to do those with Trk-B and kinase cascades. And if you'll remember, Western blots are just staining a little bit more quantitatively. And we just want to do that in order to understand how much Trk-B activation there was by measuring the phosphorylation. And then moving into tyrosine hydroxylase immunostaining, if you'll remember, Amelia explained, immunostaining kind of consists of both Western blots and immunohistochemistry, which are both semi-quantitative. So the point of that would be kind of just to quantify and visualize the activation of the Trk-B signaling cascade and tyrosine hydroxylase in both the striatum and the substantia nigra. And just as a little side note, because we are administering the Trk-B treatment later, it would be a more of a restorative model. So therefore, it would be a little bit more clinically relevant.
Yeah. And so just to clear up any confusion, these are our two primary experimental groups that we'd be looking at. So we'd be interested to see how an injection of that Trk-B would affect our MPTP model if we inject it 4 weeks later to be restorative, and then the same with our alpha-synuclein model. So inject the Trk-B AAV virus into the alpha-synuclein model 4 weeks later. So our hypothesis. So what we think is that we'll see similar results to the original DHF study. So we think this because we hypothesized that the DHF treatment did in fact work by activating our Trk-B receptor cascade. And so overexpressing the Trk-B receptor, we think will result in similar effects. So we think we'll see a recovery of striatal tyrosine hydroxylase and those dopamine synapses. We hope to see an increase in our Trk-B signaling cascade activation, but we don't hypothesize that we'll see much recovery in the substantia nigra of those already degenerated cells. So another thing that we hypothesize is we think that there might be a greater amount of recovery in our alpha-synuclein model. So if you'll remember that really intriguing interaction that stated that alpha-synuclein can kind of inhibit Trk-B, we think that maybe that relationship is why Trk-B is involved in Parkinson's disease pathology. So that's why we kind of think we'll see more drastic results in our alpha-synuclein model as opposed to the MPTP toxin model, which we think works in a different via a different mechanism from Trk-B. So here are the publication references that we mentioned today in our talk. And we just want to say a huge thank you to all of you for listening to us, for Saturday Academy and ACE, the VA Portland Healthcare Systems, and OHSU for this incredible opportunity.
Yeah, and of course, a huge thank you to our amazing mentor, Dr. Meshul, for just being such an incredible educator and for sharing his passion for Parkinson's research with us this summer.
Yeah.
Well, thank you very much. Are there any questions from the audience?
I had a question. And I apologize if you already answered this, but I was wondering with your proposed experiment where you're planning on overexpressing Trk-B, are you planning on doing that in addition to stimulation of the Trk-B pathway or just overexpression of the Trk-B?
I can take this one. So I think that we're planning on just overexpressing that Trk-B in hopes that therefore the BDNF that is present in the brain will be able to result in more phosphorylation. The reason why we're not doing it with DHF is because kind of the purpose of this study is to eliminate any potential pharmacological side effects. So that's what we're currently planning on doing. Did that answer your question?
I think so. Thank you.
Okay, awesome. Thank you.
Any other questions?
I don't have a question, but I just wanted to say this is a great presentation. And as a physician that sees so many patients with Parkinson's and someone that has a close family member with it, I appreciate this work. So thank you.
Thank you.
Thank you.
I agree. Thank you guys for a great presentation today. And we would all be applauding right now. So thank you.
Thank you.
Alrighty.
Thank you. So thank you, ladies. The next presentation is by Emma from St. Mary's Academy. She is going to be talking about improving the functionality of joints on a monoarticular quadruped robot. So Emma, whenever you're ready, you can go ahead and get started.
All right. Good morning. My name is Emma Iwaniec. I'm going to be a senior at St. Mary's Academy, and I spent this past summer interning at the Agile and Adaptive Robotics Lab at Portland State University. And today I'll be talking about improving joint functionality on a monoarticular quadruped robot. So this is a brief overview of everything I'll be talking about today. I'm going to start with a quick introduction to the lab and the work being done there. Then I'll discuss my work that I did this summer with minimizing encoder slippage and reducing joint friction with pulleys. I'll wrap up with a few big ideas and a conclusion, and then end with some acknowledgements. Okay. So the Agile and Adaptive Robotics Lab combines mechanical, electrical, and many other subsets of engineering to replicate biological locomotion using robots. The main purpose driving the lab's work is to learn more about the organisms they mimic and to apply these discoveries on a greater scale. The lab features many exceptional projects, but the one I spent the most time with was a quadruped robot, which you can see in the top corner. This robot, affectionately dubbed Muscle Mutt, is a lab-engineered robot designed to mimic the locomotion of a canine, and it uses catted skeletal features, pneumatic muscles, and an electrical-based neural network. The robot has monoarticular legs, so the muscles only span from one joint to another and never go beyond a joint like they do in the biarticular pair of legs. Some goals I had when starting this internship were to gain more experience with engineering, particularly using CAD design and 3D printing, and to observe the intersection between biology and engineering and see how biomimetic robotics can help improve our world. So as the AARL seeks to replicate natural movement, they have to collect data about the performance of their robots. And one way that the lab collects locomotion data is by using encoders. Encoders are electronic measurement tools that evaluate rotation or displacement. And so in this quadruped robot, encoders are placed at each joint to measure rotation when the robot is in motion. As flexion or extension occurs, a shaft connected to the encoder moves with the limb, allowing the encoder to collect information about the joint. However, with the original design in place here, the shaft did not rotate with the joint accurately, so the encoders could not track the movement occurring. My first project in the lab was to adjust the shaft and joints to minimize this encoder slippage. I did all my redesigns for this project using a computer-aided design software called SolidWorks. And in SolidWorks, I could create original or alter pre-existing designs using 3D modeling and then print those designs using the lab's 3D printers. I chose to focus on one joint at a time, beginning with the left scapula. And the first thing I did was adjust the shaft. I made the D profile more prominent and decreased the profile height. And I did this because a more prominent D profile will make sure that the shaft and in turn the encoder rotate with the scapula instead of the scapula just spinning around the shaft like it had been doing previously. And then after I adjusted the shape of the shaft, I then had to edit the corresponding joint holes to match the new shaft profile. I also included heat set inserts and set screws in my design. A heat set insert is a special part that can be inserted into a 3D printed hole to add threading and then house a screw. And I chose to use these heat set inserts instead of just including threads in my CAD design because 3D printed parts can be pretty finicky and there's always some variation in precision. However, the heat set inserts had their own set of challenges because they have really strict dimensioning and I had to include a tapered hole. I also added a counter bore to the top of the heat set insert holes so that it would be easier to install those. Then following the installation of the heat set inserts, in my design, a set screw would go through the top of the joint and into the shaft to add some extra security for when the joint rotates. Once I finished my initial designs, I got to print and test them. And so I printed the redesigned scapula as well as the new shaft, which I infused with carbon fiber. Though in the final version, it'll be metal for added strength. The initial round of prints did not fit together as I intended, so I had to spend some time redesigning the holes and shaft sizing before printing some more versions to test. Once I found dimensions that worked well together, I was able to transfer my design over to the other joints on the robot. The right scapula and the hips were fairly straightforward, but the knees, wrists, and ankles were unique in that they weren't open on the top, so the set screw could not be inserted vertically. For those joints, I chose to increase the length of this shaft and CAD an offshoot to house the set screw, which you can see up at the top of the slide. And then I printed out some versions of the new joints and tested fit once more until I settled on a successful design iteration. So after redesigning the encoder shafts so that hopefully accurate joint data could be collected, I turned my focus to the joints themselves. So the muscles on the robot are pneumatic and have tendons represented by these pink strings. Attachments between tendons and bones are represented by how the string is tied to the catted joints. In the original design of the robot, the tendons glide over barrel screws like the one you can see in the picture here. However, this is not ideal because it causes friction when the joints are extended or flexed. And additionally, as the tendon continuously rubs against these barrel screws, they can spin and eventually come undone. Well, this is a really prevalent issue on the monoarticular legs that I've been working on. The biarticular legs have successfully evaded this problem. This is because, as you can see in purple here, the team that developed these legs used pulleys to guide the tendons instead of barrel screws. So as I began working on this project, I took inspiration from the biarticular legs, and I made a copy of the pulley model used there and adjusted it to be wider and symmetrical. The pulley is designed to sit on top of a bearing that spins along with it, which helps minimize friction. I also had to adjust the upper piece of the joint to better house the pulley, and I did this by deepening the extruded cuts on each side and then testing the clearance using a SolidWorks assembly. Within the assembly, I could use the mating feature to test how the actual pieces would fit together in real life. I decided to completely replace the barrel screws with a custom shaft designed from scratch in order to really minimize the chances of the screws coming undone. I designed the shaft to be press fit to ensure security, but also help facilitate easy assembly and disassembly. The idea behind the shaft was that it would be inserted in the upper joint through a bearing and pulley while also housing a screw and a nut. I added a head with a hexagonal cutout to hold the nut in place while a screw could be added from the other side. I went through several iterations of this design before finding the best option and also had to account for printer tolerances when printing. There are also a few barrel screws guiding tendons along the torso of the robot, which you can see circled in pink here. These pieces were different than the knee, ankle, wrist, and shoulder joints because they have a unique shape and were oriented differently. However, by widening and realigning the extruded cuts along the side, I was able to create a design that includes pulleys along the torso as well. Though the pulleys helped reduce friction with the tendons, they also resulted in the bottom of the muscle being angled slightly forward, since the pulleys protrude more than the barrel screws initially had. In an effort to keep the muscles straight and flat, I had to adjust the upper attachments of the muscles. I did some quick calculations using the diameters of the bearings and pulleys to determine how much the muscle had shifted, and then I expanded the upper attachment by that amount. So after implementing these pulleys, I noticed that they interfered with the joint, and this needed to be fixed so that the joint could experience a full range of motion. I found a solution by moving the location of the pulley up the leg and then creating a new triangular base so that the lower joint could sit as it previously did without hitting the pulley. Now the joints are able to fully flex and extend without causing extreme friction, and the new shafts can withstand rotation without moving. Hopefully, with the changes I made this summer, the monoarticular quadruped joints will function with a higher level of reliability. This could also have an impact on the lab's ability to accurately and confidently collect data about limb rotation and minimize the effects of friction or misfit parts. My adjustments will also hopefully make the assembly and disassembly of parts of the robot easier and quicker, so future changes can be implemented smoothly. The importance of the lab's work is really evident. The ability to use biomimetic robots like the quadruped one I worked on this summer is essential for better understanding locomotion and the biological adaptations of organisms. As for me and my journey, I hope to continue exploring the world of bioengineering, and I feel empowered by the skills I have gained from working on these projects. I would like to extend thanks to all the amazing people who made this experience possible for me. First, I would like to thank my fellow interns for ensuring a supportive, cooperative, driven, and fun environment this summer. I'd also like to thank Portland State University for hosting me, as well as John, Emma, Ben, Lawrence, Joe, Jordan, Cody, and all the amazing mentors at the ARL for sharing their experience and making sure us interns were supported. A sincere thank you to Dr. Alexander Hunt for giving me the wonderful opportunity to work in his lab. And finally, I would like to thank the ACE staff, including Mara Hand and my teacher monitor, Linda Wolf, for facilitating this amazing experience and keeping everything running smoothly. And thank you all for your time.
Thank you, Emma. That was a great presentation. Are there any questions?
I do have a question, yeah. So a lot of kind of what you did in the internship from what I've seen is like problem solving with these joints and the tendons. And I'm curious, like once you encounter a problem with your design, what's kind of the first step you do in your problem solving, I guess?
Yeah, that's definitely something that happens a lot. It's very rare that your first attempt to solve a problem or create a design is going to work perfectly. The first thing that I did when I encountered stuff like that was making sure you identify whatever's going on right away. So like with the pulleys I added, seeing, okay, it's interfering because the diameter of the bearing is bigger than the barrel screw was. And then it's almost trial and error from there, being open to trying lots of new creative solutions and seeing if you can find one that works. And also asking for help and getting input from all the other amazing people in the lab is really helpful for tackling those problems.
Awesome, thank you.
Any other questions?
Emma, what do you think is the most important thing you learned this summer?
Wow, that's a big question. I mean, I would go back to the problem solving, being able to look at a problem and come up with a conceptual solution and then turn that into actuality has been amazing. And getting lots of practice with the design cycle, reworking things when they don't work out, learning how to move forward and adjust when things don't work perfect the first time. And then just getting to work with so many amazing people on such cool projects has been incredible.
Anybody else? Okay, well, thank you so much, Emma. I appreciated that. Thank you, Emma, and the team from Portland State. Next up, as a fellow intern at Portland State, we have Sarah. And Sarah Woods, she is from Wilson High School. Her presentation is design of Gastrocnemius pneumatic artificial muscle attachments on a Biomimetic quadruped robot. Primary mentor was Dr. Alexander Hunt, and again, she was at PSU. So Sarah, when you are ready to go, you can go ahead and present for us.
And my video's not started. Wait a minute. Okay. Sorry. Oh my gosh. Okay. So hello, my name is Sarah Woods, and I'm a rising senior at Ida B. Wells Barnett High School, formerly known as Woodrow Wilson High School. And I worked at the Portland State University Agile and Adaptive Robotics Lab under Principal Investigator Dr. Alexander Hunt. Today I'll be discussing the design of pneumatic artificial muscle attachments on a biomimetic quadruped robot. So today I'll be discussing, I'll be going over SolidWorks, MATLAB, wiring, professional development, and then just go to acknowledgements. So we began this internship with a fair amount of SolidWorks, which is a computer-aided design program. We completed about 28 tutorials ranging from parts and assemblies and design tables to assembly visualizations and animations. We then applied that learned knowledge to our future tasks. My first task was improving the lower biarticular muscle mounting. The lower biarticular muscle is also known as the gastrocnemius muscle, which is the calf muscle that flexes the knee and the foot. And because it flexes both of those joints, it makes it a biarticular joint rather than only crossing one joint, which would be a monoarticular joint. That muscle was previously attached by two tendons, which were represented in lab as strings, and then are represented by pink lines on the CAD model. But being attached by the strings meant that the muscle would be out of plane, and we want it to be in plane with the muscle. Otherwise, it can lead to force being applied at a strange angle. So our solution was to pin it on both sides to limit the range of motion and fasten it in plane with the muscle body. So these are the different revisions of the knee joints. I was able to get a lot of them done, fortunately, because we have access to 3D printers, which is nice. And on the right, we had the first design, which is quite bulky, and it didn't end up working out because it's got the backing to it around here. So it didn't have the degree of rotation to be able to spin backwards. And then here, it was hard to get the pin and post system in also. And then the middle one, we have like two little hands sticking out. But what that does is it rotates the muscle 90 degrees counterclockwise. And that didn't allow the proper degrees of rotation either like along this angle or along the angle like here. So it just didn't end up working out. And then on the left, we have the other knee joint. In the middle, you can see that there's a broken off piece because the support wasn't strong enough, and it also didn't allow for the pneumatic cap to be able to swing in there. So that led us all to the final knee joint, which has a support along here and then a support here, which are thicker so they wouldn't break like that other one. And then it's got a pin post system here, so it is able to swing in the desired degrees of rotation, and it keeps the muscle in plane with the muscle body. So then we moved on to the ankle, and because the top of the braided pneumatic actuator, or BPA, which is the name of our artificial muscle, because it was pinned as it was, there's a new need for a pulley, so it could lead around the tendon, so it could lead the tendon around the pulley to a further pinning point on an ankle. So in the cyan box, you can see the pulley as an extension on the pre-existing axle, which is helpful for strain calculations, which we mentioned later. And then in the red oval and box, you can see the pre-existing ankle, which is in the CAD model, and then the new piece, which was 3D printed on the left in the red box. And there's a new hole in it to add a heat set insert and then a screw to be able to pin the lower tendon. So then we moved on to the calculating strain to be able to find that pinning point on the ankle. And strain is how much the muscle contracts, essentially. And we wanted to minimize that quantity. So we used a strain calculation program on MATLAB, which is written by our mentor, Emma Kornacek. In the left, you can see the section of the code where you can enter your custom pin and pulley coordinates, which with respect to their axles they're nearest to. So that's when the pulley being directly as an extension of the ankle axle comes in handy, because it means it's as close as it can get to the axle with still being like respective to the 3D printing desired values for certain like load bearing prints. And then on the right, we have the output when you run the program, with the yellow boxes being where you can customize those values. So we can change the hip, knee, and ankle rotations, and then we can change the actuator lengths. And in this situation, we didn't change the actuator lengths because we were just testing different coordinates with all the same like area. And then that led to the output here, which is the initial position before the hip, knee, and ankle degree rotations on the left, and then the position afterwards. And that's really helpful that we did it through MATLAB, because it's a programming language that's for numerical computation, but it's just generally good also at these types of plots and like images, so you can see stuff better. And then at the bottom, you can see the strain output for the strain values at the different points, and the one we're really looking at now is at the ankle extensor. And then this is the data we put in a spreadsheet to look at the different coordinates to find the best one. But essentially, we learned that with the pulley and knee pin location, it wouldn't be possible to get under the desired level of strain, which is around 0.17 or 17%. And the pulley and knee pins were already about as close as they could get with the pulley being directly as an extension on the ankle axis, and the knee pin being shortly above the axis or the axle. However, the ankle extensor strain still remains under the manufacturer recommended level of the strain for these muscles. So we just picked a random point and used that. So this is the final product. We have on the left, the new ankle joint with the white taping where the hole would be, and then where the hole is with heat set insert. However, the heat set inserts haven't come, so we haven't been able to finish that. And then we have the purple box with the new pulley directly on the axle. And then the green box is representing the muscle we've been working with. So as you might imagine, having two pink strings attaching it on each side might not keep it the most in plane. And then on the right, we have the new knee joint with the pin post system and the supports. Another task I worked on was the ground contact sensors. We wanted to add ground sensing capability to all 4 of the feet on both the monoarticular and biarticular quadruped. It's necessary feedback just in general for walking. However, it's more important for changes in elevation. In the bottom left corner, you can see the foot design for the biarticular robot, which we want to implement into the monoarticular robot. And you can see the 2 yellow lines representing places where we could place the strain gauge, which is pictured above the foot. And then the line on the bottom, while it works, it would get a lot of wear and tear, constantly like hitting the ground and stuff, which isn't good considering how sensitive these strain gauges can be. So we wanted to do the left area, the line on the left, because it would get less wear and tear from constantly hitting the ground, but it would still be able to give us the necessary, like the values we want, which is basically just a, are we in contact or are we not? Although we were thinking of possibly thickening the foot a little bit, so it's not as like hypersensitive, because right now it's pretty like flexible in a way. And while we could technically determine these values ourselves through like our already received values in muscle pressure, it's better to get the feedback ourselves if possible. So then I worked on wiring the monoarticular quadruped. So on the right side of the robot, you can see the encoder wires, which we want to connect to the circuit board, which is in the bottom right corner. And then we have the power wires or pressure sensor wires coming out of the middle of the robot with some power wires that need to be like cut back, stripped, then extended and spread through the robot and stuff. And so you can see the process for that included grouping them over here, because each encoder has 4 wires, a red, green, blue, and black, and the red and the black are the power and ground. And then the blue and green are for other purposes. And so we extended them all so they could reach the circuit board, and then we twisted them together for neatness. And then the pressure sensor is similar, so I'm not going to divulge too much on that, but we can see it before and after. So you can see that the wires are just like sticking out and not long enough to reach the circuit board, which would be on a table somewhere like over here. And then the pressure sensors just aren't ready at all, but here you can see the pressure sensors are like kind of like thread through the robot, and then they go through here, out here, which is harder than it looks considering the robot channels aren't the biggest, and they're already fairly full with a bunch of other wires. So yeah, that finished up pretty nicely. And then I also worked on Engineers Without Borders climate research. So the next Engineers Without Borders trip will be to a community in Rwanda called Kanemba Botwe. And the objectives of the trip include designing and building a new water supply for a rural Rwandan town. It's expected to include catchment, extensive piping, and collection kiosks. In the bottom right corner, we can see the photo of where the town is in respect to Rwanda. And then we can see in the middle photo where Rwanda is in perspective to all of Africa. And so there's it's the top of those 2 little countries, with Burundi being below it, Democratic Republic of the Congo to the left, and Uganda above it. And then on the far left, we have like an aerial view of the like town. And what we learned basically is that there's less rain now, and then they have their like normal 4 seasons, but we weren't able to get like a lot of data because there's not a lot of like sources around there for us to be able to get the data, unfortunately. Another opportunity we got was to attend the Living Machines conference, where we got to hear from speakers from Asia, Australia, Europe, and North America. We participated in the main plenary talk, as well as 3 workshops, including bio-inspired locomotion, invertebrate robotics, and bio-hybrid robotics. So that was really cool, and we basically just got to see how biomimetic and bio-hybrid robotics is doing in like different places and what other people are working on. And then finally, I'd like to thank Dr. Alexander Hunt for making this possible, as well as Portland State University and the Agile and Adaptive Robotics Lab. And I'd like to thank Saturday Academy and our teacher monitor, Linda Wolf. I'd also like to thank our mentors, Cody, Ben, Emma, Lawrence, John, Jordan, and Joe, as well as my fellow interns, Aidan, Emma, and Jeffrey. Thank you.
Thank you so much. That was fantastic. Wow. Are there any questions from the audience?
Yeah, I wanted to ask what one of the biggest setbacks or challenges you experienced this summer was.
That's a good question. I'd say, that's actually a really tough question. I'd say that one of the hardest things might have been the designing at the beginning, because I was newer to using SolidWorks as a program in general. I'd been more used to using like other CAD programs, and then just like trying to get like a solid design that didn't look bad either. Yeah.
I have another question. Just clarifying from someone who doesn't know much about robotics, you mentioned ground wires and power wires. I'm just curious what ground wires are.
So ground wires are like what, like push out the electrons basically, so you don't get like electrocuted when you're like, okay, so like computer wires, when you plug it into the wall, a lot of them have three things, the two normal ones, right, and then the one below, and the one below is a ground wire, and that's just for like, it's like, so there's not too much like, the way I understand it is there's just like not like a lot of power going through it then, or so it's like in a dangerous way, it just like it exports all the electrons just out. That's the way I understand it. Sorry, that's not the best description though.
That's great. That totally makes sense. Okay, thank you.
Anyone else? Okay, well, thank you so much, Sarah. That was a great presentation. Great project that you did. So at this point in time, kind of up to our room, what we'd like to do. We could take a short break right now, or we could go on with our next presentation. Let's see, could I get some responses from everybody? Would you like to take a short break before we start? Maybe like a 5-minute break? Or would you like to just continue on with presentations? Okay, so thank you for the 2 responses. Let's just take a short break before we get started. So let's plan on a 5-minute break, maybe 6 minutes, just to get us back here on time. So see you guys back, and we'll get started. Let's see, at about 10:48, according to my computer. So I'll see you back then. Thank you, everybody. Hi, everybody. I hope you had a great break. We have 4 more interns to present, and I am hoping everybody is back. It's always difficult to tell. Our next presenter is Aidan. He is from Grant High School. He is going to be talking about finding the relationship between force and pressure in pneumatic muscles. His mentor was Dr. Alexander Hunt, and he was at Portland State University. Aidan, are you here and ready?
Oh, yeah, I'm ready, yeah.
Okay, awesome. Thank you so much.
Okay, let's go. Okay. Well, here we go. Okay, so I'm Aidan Vogt. I got a grant. This summer, I interned at Portland State University in the Agile and Adaptive Robotics Lab. And today I'm going to be talking about finding the relationship between force and pressure in pneumatic muscles. So these are the muscles we used in our robots at the lab. We used them since they are more accurate in representing real animal movement, since they contract similar to how real muscles contract when we pressurize them. Like this air comes in from one of these tubes, pressurizes it, and then it contracts like this, and that'll mimic a muscle movement. But a big limitation of these, as you might be able to tell from the pictures, is that it doesn't contract very much. Real animals have much more contraction in their muscles. So to compensate for that, we use much larger and much longer muscles than what are found normally in animals, but they do a much better job of mimicking animal movement. So in order to measure the force and pressure, as we would want to do if we want to know how much pressure to pressurize them in order to get a certain force, we need the tools to do so, which are here. On the left is a pressure sensor to measure the pressure. The tube here, there's a second tube on this muscle. This is for the air. And then another one goes directly into the pressure sensor to read the pressure. And then we have a load cell, which measures the force that the muscle is providing. When you pull on these two overhangs right here, the pressure is or the force is then measured and sent through this wire. But since the signal here is a bit too low for what we're going to be using, which is an Arduino to read the data, we send it through this amp to make it more readable, and then it goes into the board, which then allows us to read the data that we need. So, but before we can use these, we do need to calibrate them to make sure we're getting accurate readings. So to do that with the pressure sensor, we hook it up to the Arduino, where we can then read a value between 1 and 1023, the values in this table, which we can get when we pressurize the tube to a known pressure that we know, like all these pressures right here. And then once we have all of the analog values that correspond to these, we can plot them, find the line of best fit. And once we have that equation right here, we can then work backwards. If we have an unknown pressure, we can then take the analog value that we're reading from the board, plug it into the equation, and get the pressure of the muscle that we need for when we're finding out all the tests we're going to be running. We're going to need to know pressure. That's how we're going to find it. It's a very similar process for the load cell, except it's a bit different since the load cell is going to be in the actual setup. So that means it's going to be holding things like its own weight. It's going to be holding other things. So to do that, we need to zero it in its resting position. So in this example, the resting position is it hanging horizontally between these two strings. It's actually hanging between, this is a limb that's going to be pulling outwards to pull the load cell to get the force reading. So that's the zero position. And then after that, we can start doing a similar process as the pressure sensor by adding different weights to get how it scales upwards with higher weights to know, again, the equation to find the force when we get a certain value reading on the Arduino, which allows us to read both force and pressure, which are the two stats we're going to be measuring in the tests in the future, which I will going to be explaining later. So now that is all the way. We want a convenient way to read the data. So we have the board right here. It's hooked up to both the tools, but we're going to want to store it in an easy way. So the way we do that is by using MATLAB, which is a program that allows us to easily store and display the data in a lot of useful ways. And so the way we do that in this is with a table, or not a table, this is a graph that shows the force and pressure over time in seconds throughout the duration of the test. So we have it set up so that the Arduino will receive a command from MATLAB telling it how much data to collect, and then it will start collecting the force and pressure. The blue line is force, red line is pressure, and then it'll just plot it on a graph that we can see pretty easily, as well as putting it in a nice table with all the data points that we can put in like a spreadsheet and compare very easily, as well as putting it in a table of some basic stats about it that we can easily just read if we want to see a brief overview of how the test went. So right now, that's how that works, but we're going to also need to pressurize the muscles in order to start the tests. So the way we do that is by using these valves right here, which is connected to the pressure with this tube right here. These tubes are connected to the muscle, one of which is to pressurize, the other one is to depressurize, as like an exhaust pipe, if we want to depressurize the muscle quickly. And then this one is connected to the pressure sensor. But in order to turn on these valves, we need higher power than what the board can provide, since the board can only do around 5 volts of output. So we use a 24-volt power supply here. And since 24 volts is way too high for this board, if we connected them directly, it would burst, it would explode. So we connected it with some transistors, which basically act as switches for the Arduino to be able to turn on and off with low power, to safely turn on and off the valve when we need to, without subjecting it to the high voltage of the power supply. So right now, the setup in this picture is able to receive a command from MATLAB, open the valve, and then start collecting data and storing the data in MATLAB to be able to run our tests. Yeah. Another thing we might want to do is not have the force be always fully on all the time. So just to be able to have more control over how quickly the muscles contract. So the way we solve that is by pulsing the pressure on and off like this, like by closing and opening the valves. Like here, the valve is closed and it's open, close, open, close, open in quick succession, to be able to extend the time it takes for the pressure to fully go into the muscle and for the muscle to fully contract, as well as how long it takes for the muscle to re-extend to its resting position on the other end. And by being able to vary the times that these are pressurized and unpressurized, we can have great control over the muscle and how quickly it contracts. So that's very helpful for our testing, and we're going to be using that in the future. Another thing we had to test was the caps on the muscle. As you can see here, we have to have 3D printed caps to connect to the pressure, as well as to keep the air in. So there's multiple designs here, a lot of them, and we have to test a couple things about them, such as how quickly we pressurize the air, or how quickly we pressurize the muscle when air is put into the cap. So how we do that is with this graph here, we can have over time the pressure as we put in the air, and we can compare each cap, how quickly they reach the max pressure, or the pressure of the air that we're putting in, which in this case is around 620 kilopascals. And then we can see, we can compare them and see which one fits our needs the most, because we don't want a cap that fills in necessarily too fast or too slow. We want to be able to know about how quickly our thing is going to pressurize, and then use the cap that best fits our needs or the muscle that we're going to be using. And then another aspect of these that we have to test is leakage, because these aren't perfectly airtight. There are some areas for the air to escape. So by measuring how long it takes for the air to reach a certain pressure after it's been inflated and pressure is held and the air starts leaking out slowly, we can see which caps do a good job of holding in air and which ones don't. So we can maybe see some patterns and design our caps in a way that better hold the air, and maybe make them so that they fill at a time that we want them to, as well as being useful in other ways, I guess. And then here are some of the tests we've been using these on. This is a test that it just has a muscle directly linked up to a load cell. This is how we get the data like this, where it's just force and pressure of the muscle itself. It's used mostly with the pulse pressure right now, so we can see how different frequencies of the pulsing and different lengths of pulses affect the force and pressure and how long it takes to inflate and deflate over time. That's what this is used for. And then we have another test, which involves the knee. This is a 3D printed knee built to replicate sort of a human knee. And the test has a muscle on the outside. When this muscle contracts, the knee expands or extends. And then we can measure the force between this arm and the tip of the knee. to see how much force the knee is able to provide on the load cell. But a slight issue with the setup is that since we want to measure at different angles on the knee, say we want the knee to be at this angle, and then the arm would be at a different angle, and the load cell would be here, it means that the load cell is not going to be at a 90-degree angle or even like the same angle throughout the entire range of motion that we're going to be testing, which is an issue if we want to compare the data, because if we have a different angle every time, it's not like we can really easily get much useful comparisons out of it since they're all going to be different. Luckily, we can you can do some trigonometry to find the component of it that's perpendicular to the arm or to the leg muscle here to find it. But we do have to know this angle, which isn't the easiest thing to find accurately. But if we do, we can in the future maybe be able to find this angle and measure the forces at different angles of the knee. And that's pretty much what I did this summer. I'd like to thank everyone, including Dr. Alexander Hunt, for letting me work in his lab. It was really fun this entire summer. All the mentors, Ben, Lawrence, Cody, John, Emma, Jordan, and Joe, for teaching me so much and allowing me to do all this work, as well as PSU for having the lab, and Saturday Academy for letting me do all this stuff. It was really cool. Thanks for listening.
Thank you, Aidan. That was a great presentation. What a great experience for you guys to all be working in the same lab. Does anyone have any questions for Aidan right now?
I think my favorite part was learning to connect the valves to the setup. I think it was cool to be able to program a certain amount of pressure and seeing the actual muscle inflate at the right times. That was cool. Pretty satisfying when it worked properly. Not very satisfying when it didn't, but at the end, that was fun and probably my favorite part.
Anyone else?
Yeah, probably I would want to be an engineer. Probably. I'm not exactly sure the specifics about what exactly I want to do, but definitely is something I'm very, very heavily considering in the future.
Okay, well, thank you so much, Aidan. And appreciate your presentation. So thank you again. And it is time for one more student who was in Dr. Hunt's lab. There were 4 interns from ACE. And the last one who's going to be presenting is Jeffrey Burt, who is from Catlin Gabel School. Presentation title, Design and Fabrication of Custom Testing Equipment for Legged Robots. And he worked at Portland State. So Jeffrey, are you ready?
Yes, I'm ready, but it says that I cannot screen share due to being a participant. So I think I might need to be made a co-host in order to do that.
Sorry, I thought I made you a co-host. Hold on.
Oh no, it's fine. My Zoom crashed earlier in the meeting, so I had to rejoin. But
oh, okay.
no, I think
Here
that's
you
on
go.
me, not you.
Should work now.
Thank you.
Yes, there we go. All right, and can everyone see this and hear me well? Yes. So yeah. Again, my name is Jeffrey Burt. I am currently a rising senior at Catlin Gabel High School, and I interned at the Portland State University Agile and Adaptive Robotics Lab. And I'll be presenting today on the design and fabrication of custom testing equipment for legged robots. And so just a quick overview of what I will be discussing today. I'll start off with just a little description of what the lab does and why their work is important. Then I'll go into my work area, which again is the testing of a bipedal robot that uses artificial muscles. Then some background on the testing setup that I was using. Talk about these sensors that I added to the testing setup, and then some electrical background of the testing setup, including printed circuit board designs and some op-amp support circuitry for these sensors. Finally, do acknowledgements and questions. Yeah, so the Agile Adaptive Robotics Lab, and their goal as a whole is to closely study how animals move and then apply that learning into mechanical robots. So this work is incredibly important because, in case you haven't noticed, in general, animals are very far ahead of robots in terms of movement. So for example, as humans, we can walk up stairs, or we can walk along uneven and rough slopes with almost very little thought, and it's very easy for us. But for a robot, something like that might be impossible. And so what the lab is trying to do is trying to see how animals move and then apply that and make it so that robots can move at the same level using the same tricks, so to speak, that animals use. And they do this in a few ways. Firstly, through control systems. The lab uses computational neural networks, which is a fancy way of saying they simulate the animal's brain to basically tell the robots how to move, so where to put your feet and where to move the legs and such. Then they also use mechanical systems that are very closely modeled off of animals and animals' mechanical systems. So for example, if you're building a robotic arm in the lab, they might try to build the arm to be very similar to a human arm. So they would have 3 printed bones where humans have bones in their arms, and they'd have the joints move in similar ways to humans' arm joints. And then finally, the last piece of the puzzle is the actuators that the lab uses. So unlike most robotics, which uses electric motors to move their robots, the lab uses braided pneumatic actuators as artificial muscles. And I'll discuss these a little bit later, but these essentially are the last piece in the puzzle. They allow the control system and the mechanical system to work very similarly to animals' systems, and they just tie everything together. Yeah, and then so my work area was improving the testing setup for a bipedal robot knee. And a bipedal robot is a 2-legged robot. So we have this design of a knee that is modeled after a human knee. It's eventually going to be used in a 2-legged walking robot. But before we can do that, we need to test the knee model to see how well it works, if we need to make any changes to it, especially how it relates to the artificial muscles, because we know how the artificial muscles work. We know how much force they'll produce at different length and different pressures, but we don't really know how they'll interact with the knee model itself. So what we're doing is we are just using the artificial muscles connected to the knee, activating the muscles at different pressures and forces and such, and then measuring the force that the muscles exert on the knee. And this is important because it allows us to essentially validate the knee model that we're using. So the mechanical model we're using, we can compare our experimental results with theoretical results and even with results from actual animals to see how good of a job we did at designing the knee and if we need to make any changes. And so we've heard the presenters before me have mentioned the braided pneumatic actuators, and these are very special and very specialized components that work very similarly to a Chinese finger trap, actually, if you've ever used those like, when you were younger. So if you remember for that, when you pull out, they get narrower, and then when you're pushing in, they get wider. And so what the muscles are doing is they're putting essentially a bag that you can fill with air in the center of these Chinese finger traps, so to speak. And when they fill the bag with air, that expands and increases the diameter of the muscle, which causes it to contract. And that's the contraction movement from the muscles. And there's a few things that let us use these as artificial muscles. Firstly, they're compliant, which is fancy robot speak for saying they're basically like springs. So for a human arm, if you push it, it will move out of the way. But for a traditional robot, if you push it, it will just not care. It will not notice you're pushing on it. So what these do is when you push them or when you pull on them, they'll absorb the energy by moving and deforming or stretching. And then finally, they're also linear movement. I mentioned before how most traditional robots use rotational movement. So that's spinning around for motors, but these just go in one direction. So they're pulling a string or something, linear movement, just like human and animal muscles are. And here's a video of one of the muscles contracting. Yeah, and so moving on to what I did personally, so the testing setup, as Aidan mentioned before me, we need to know the angles and the positions of everything in order to be able to quantify the forces that the muscles are exerting on the knee. So we need to know the position of the knee itself, and in order to do that, previously what we've been doing is we've been using a camera setup. We've been taking pictures and using some specialized software to go through for each picture and measuring the angle of the knee that way. But this is a bit slow for someone needs to go through for every single test and spend a few minutes finding the angle. It's a bit inaccurate too because cameras are a bit noisy and such. And so in order to counteract this, we are adding, or I added an encoder to the knee. An encoder is a rotational position sensor, which will hopefully allow us to get much more accurate data much quicker. Excuse me. So you can see. There we go. Yeah, so here is the encoder we added to the knee. This was actually a fairly complicated design process. I don't have time to fully go into it, but put briefly, the knee itself is a very complicated mechanism that's already very compact, so it's made to be as small as possible, and that makes adding new things to it difficult. And in order to add this, we had to actually convert the rotary axle that the knee travels on, or one of the linkages in the knee travels on, from a dead axle to a live axle. So that means it rotates with the knee. When the knee rotates, the axle will also rotate. And this is needed because we can't fit an encoder down here in this area because it's too tight. So in order to transmit that rotational data, we need to move it along the axle. And that design process was also fairly difficult because, again, it's very space constrained. And secondly, in order to constrain the shaft here and keep it from just falling out, you need to really tighten it down. However, this adds a lot of friction to the model, which is bad because you want the model to work as closely and as closely to a human knee as possible and as smoothly as possible. And so in order to counteract this, we're adding multiple bushings throughout the process, or throughout the mechanism. Another thing I worked on was creating a custom printed circuit boards to support the testing setup. And these essentially allow us to be more repeatable in our testing setup. So rather than for each different setup we're trying to make, previously what we were doing is we were going through and making manually creating a circuit in a breadboard, which is an electronic device or electronic circuit making tool. But with a printed circuit board or PCB, what this allows us to do is it allows us to eliminate some human error in the process, so you don't accidentally put the wrong thing in the wrong place or plug the wrong thing in the wrong place. And it also allows us, again, to be more repeatable, so the circuit will be the same every time. And these were designed in Altium Designer, which is a industry standard PCB design software, and they are made to be very easy to assemble using through-hole connections. And oops, sorry, here we have the schematic of the PCB. So this is just a circuit diagram essentially, and then that is turned into a PCB model in the computer, which is a time-consuming process as you need to individually route all the connections in the schematic here, and they can't cross over each other. So you have to be very careful in the placement of components in order to make sure that you can actually properly route the board and connect all your components inside the circuit board together. And then this digital model is then turned into an actual real life circuit board, which we can then add components to, solder things to, and used as a functioning circuit board and as a functioning circuit. Excuse me. And then finally, I also worked on the electrical circuitry to support the sensors that we are using. So we're using something called op-amps, or operational amplifiers, and these are needed for the sensors because, for example, the load cell has a extremely small output that's quite difficult to read for our microcontrollers, which it's for the people who know this stuff, it's about 15 millivolts for the entire range of the load cell. And that's quite difficult to read because, again, that covers the entire range of the about 1,000 pounds of the load cell. So a difference of 1 millivolt might be a difference of a couple hundred pounds. And so in order to make it easier to read, we're using amplifiers. So we're amplifying the signal so that instead of 15 millivolts, it'll be on the tune of 1.5 volts or so. And again, I'm using an op-amp to do this, and this was less so because op-amps are easy, more so because it's very easy to change the circuit quickly. Whereas for the traditional amplifier, if you want to change the gain or amplification ratio, you need to go and buy a whole new amplifier. For an op-amp, that's as simple as just swapping a few resistors around. And on the right, I have a picture of the testing setup I use for an op-amp. You can see up in the oscilloscope, which visualizes voltages over time. The yellow signal there is the actual signal that I'm testing with, and then the blue signal is the amplified signal with the op-amp. So as you can see, it's higher than the yellow signal, and I have a video showing how the op-amp can amplify signals of different amplitudes. Yeah, and then so I have lots of acknowledgements to make. Special thanks to Dr. Alexander Hunt for inviting me into his lab and letting us work over the summer there, as well as Ben Boland for being our direct mentor and answering all my questions, no matter how many I had, as well as the other members of the Agile Adaptive Robotics Lab, including Cody, Emma, Joe, Jonathan, Jordan, and Lawrence, the other ASE interns, so Emma, Aidan, and Sarah, and then the ASE staff, Linda Wolf. Yeah, so thank you, everyone. Any questions?
I think I'll steal Ben's question. Do you want to be an engineer?
Yes, though I'm not exactly sure what type. So this internship was actually really helpful in that regard. It gave me a chance to explore a lot of different areas of engineering. So yes.
Mm-hmm.
So Ben asked, with the op-amp video, were you squeezing it to get the blue signal to increase? I think by squeezing it, you mean kind of pushing the boundary of the op-amp's voltage max. Not particularly. I was amplifying by about a gain of 33 in that video and using only 1 volt with the supply voltage of 36 voltages. So we were close to hitting the threshold where the op-amp, physically squeezing it. No, I was not. I was changing the voltage of the signal that I was amplifying. So the op-amp would then, if you're amplifying 1 volt, 33 times of 33 volts versus you're amplifying 2 volts, it is 66 volts. So just increasing the signal will increase the amplified voltage as well.
Anyone else?
Ben just asked, is there anything I wanted to work on that I didn't get a chance to do? I think learning some control theory would have been really fun over the summer, but I have other ways to do that and I still have a lot of fun. So yes and no.
You said that you had to convert a dead axle to a live axle to allow for kind of more space for I believe the encoder. Were there any like negative effects of having to change that like made the knee less representative of a human knee?
A bit. So with a dead axle, what you're doing is you're having the bearing or the friction reduction devices in the actual moving component. Whereas you're only having the movement from that moving component interact with the static parts of the assembly in one place. So you only need one thing to reduce friction. However, with a live axle, that entire axle is going to be moving. And in order to accommodate that, that axle is going to interact in a lot of places in order to be fully defined. So you need to have multiple friction reduction things like bearings. And bearings, though good, they're not perfect. So a slight downside is that you'll have a little bit more friction, but it's not too much.
Thank you.
Yeah, good question.
Anyone else? Okay, well, thank you for a great presentation, Jeffrey. Looks like lots of hard work this summer. Thank you so much for presenting. That was great. So let's go ahead and go on to our next presenter. And I might be mispronouncing your name, so please correct me. Dania?
Correct.
Okay, oh my gosh, good. So Dania is a student at David Douglas High School, and her presentation title, How High Will They Go? Investigating Methamphetamine Consumption in a Mouse Model of Binge-like Methamphetamine Intake. Her primary mentors were Dr. Tamara Richards and Dr. Cheryl Reed Williams. And she did her research through the VA Portland healthcare system. So if you are ready to go, we're ready to hear about your summer.
Okay. Hi everyone, my name is Dania Khalid and I'm from David Douglas High School. And this summer I interned at the VA at the Phillips Lab. And my presentation title is How High Will They Go? Investigating Methamphetamine Consumption in a Mouse Model of Binge-like Methamphetamine Intake. So according to the National Institute on Drug Abuse, in 2017, there were 964,000 people aged 12 and up who were dealing with the methamphetamine use disorder, a 40.9% increase from the previous year. Methamphetamine is a highly addictive stimulant drug that once developed, it works to devastate its victims' lives. For instance, in a study conducted with 350 methamphetamine users, it was found that 61% of the sample relapsed within 1 year after the treatment. The most common use of methamphetamine can be described in a binge and crash pattern. When people consume methamphetamine, their pleasure and reward centers are activated. However, the high only lasts for a short period of time. In fact, methamphetamine pleasurable effects are gone before the methamphetamine concentration in the blood can significantly decrease. This leads people to continue consuming methamphetamine for several days without any food or sleep. And then after a couple of days, the body can no longer stay awake, hence the users go into a crash period. Thus, to tackle this persistent issue, we need a binge model to replicate these binge consumption behaviors, which could possibly lead us to discover neurobiological factors that impact methamphetamine intake for possible future treatments. Before I tell you about the binge model, I need to tell you about a gene that affects methamphetamine intake. This picture depicts its receptor called the traceamine-associated receptor 1, also known as TAR1. In mice, there are two different forms of TAR1 or TAR1 alleles that each are known to impact methamphetamine drinking differently. The common allele is TAR1+, also known as the wild-type allele because it's found in most mice. The wild-type allele encodes a functional TAR1 receptor, and mice with a functional TAR1 avoid consuming methamphetamine. The other allele is only found in the DBA/2J mouse strain. A single nucleotide mutation occurred within TAR1 in that strain that encodes threonine to a proline mutation at amino acid position 77. This allele has been named TAR1M1J. This allele encodes a non-functional TAR1 receptor, and mice who possess this allele consume high amounts of methamphetamine. The importance of this information will become clear shortly. The TAR1M1J allele and its importance to methamphetamine intake were discovered by the Phillips lab in a selective breeding project. A B6XD2F2 population was tested because B6 and D2 mouse strains were known to consume different amounts of methamphetamine. And so by mixing up their genetics via interbreeding, the Phillips lab could map the location of genes that impact methamphetamine drinking. This figure here illustrates how selective breeding is done. The methamphetamine drinking protocol the Phillips lab used consisted of giving the mice 1 bottle of water and 1 of methamphetamine. And then the high extremes of the methamphetamine intake data set were put together to produce their low offsprings in selection generation S1. The low methamphetamine intake extremes were also brought together to produce their low offsprings in S1. This continued until S3, where we can see the offsprings methamphetamine consumption graphs have separated further. This separation indicates that the selective breeding was successful and that there are significant genetic factors that influence certain mice to drink high amounts of methamphetamine and others to drink low amounts of methamphetamine. And so then by comparing the low and high extremes genetically, the Phillips lab discovered a difference in TAR1, where the low extremes had a wild type allele, while the high extremes had the 2 copies of the TAR1M1J allele. After the TAR1M1J mutation discovery, the Phillips lab wanted to study this in a binge model to see if the different TAR1 alleles are equally important in a procedure that results in higher methamphetamine intake. They also wanted to identify other genes that modify the effect of TAR1 mutation and perhaps reduce methamphetamine intake. In other studies, it was found that by increasing the number of methamphetamine bottles offered to the mice consumed more methamphetamine and simulated a binge consumption behavior. In particular, a 4-bottle choice protocol with 3 bottles of water and 3 bottles of methamphetamine and 1 of water was found to be extremely effective in increasing methamphetamine consumption levels in the mice who possessed the TAR1M1J. And so using this 4-bottle choice protocol, the Phillips lab performed selective breeding using the general method I described before. Their hypotheses were that the TAR1 impacts binge level methamphetamine intake and that there are genetic modifiers of the TAR1M1J genotype that affect methamphetamine intake. To summarize the creation of the binge model, C57BL/6J and DBA/2J mice were crossbred to create an F1 offspring population. The F1 were then crossbred to create an F2 offspring population. Then the mice were genotyped, and those of the F2 who possessed the TAR1M1J genotype were phenotyped for methamphetamine intake using the 4-bottle choice protocol I talked about previously. Mice that had high methamphetamine intake were used as breeders for the high methamphetamine binge line, and those who had the lowest methamphetamine intake were used as breeders for the low methamphetamine binge line. If breeding is successful, this would indicate that there are genes that modify the TAR1M1J effect, and additionally, the F2 mice who possess the TAR1++ genotype, they were used as the methamphetamine avoiding control line to compare to the high and low binge lines. This graph here shows the total methamphetamine consumed at each concentration of methamphetamine that was offered to the mice along the y-axis for the S4 selection generation of the binge model. Concentration is along the x-axis. Here we can see that the high and low binge lines total methamphetamine consumption significantly increased on concentrations 40, 80, and 100 mg per liter, indicated by the plus symbols. We can also see that the high and low binge total methamphetamine consumption significantly differed from each other at every concentration, indicated by the star symbols. This graph here shows the total methamphetamine consumed combined for all concentrations for each selection generation, shown as S0 through S4 along the x-axis. Here we can see the original population, the F2, graphed at S0, have drank about 60 mg per kg of methamphetamine on average. Immediately after, we can see that the high and low binge lines separate drastically. The low binge for S1 have consumed about 59 mg per kg of total methamphetamine on average. However, the high binge of the S1 have drank about 90 mg per kg of methamphetamine on average. We see this separation again in S2. However, for S3, the low binge dramatically increased their consumption of methamphetamine. This selection was during the COVID-19 shutdown when the Phillips lab had to relax selection and randomly breed within line for 1 generation. Fortunately, as they moved on to S4, the phenotypic difference between the high and low binge lines returned. And so the purpose of my study this summer was to test the final selection generation of methamphetamine binge drinking mice for two-bottle choice with escalating methamphetamine concentrations. So let's determine whether the same genetic modifiers of the TAR1, M1J, and 1J effect impact both the two-bottle choice and the four-bottle choice methamphetamine intake. In my experiment, 66 mice of the 3 different mouse lines were used, 23 high binge, 22 low binge, and 21 control binge. Different sexes were also used to investigate another potential biological factor that could affect methamphetamine drinking behavior. However, we found no sex differences in my results. To start this experiment, all mice were allowed to acclimate to the no light cycle in the selected room 2 weeks before the study began. On day -1, I weighed and isolate housed the mice. I gave each mouse 2 25 ml graduated cylinders of tap water. I also gave the mice food, which they had access to all the time during the study. And most importantly, I read fluid levels at 9 a.m. and 3 p.m. Fluid readings have been scheduled at these times for every day of the study because mice are offered methamphetamine for 18 hours each day rather than 24 hours, which has been shown to increase their methamphetamine intake. On day 0, I read fluid levels again. I refilled liquids as needed after each reading. On day 1 of the study, I weighed the mice. The weighing of the mice occurred every other day. I then removed the 2 water tubes. Instead, I placed 1 new water tube. And then at 3 p.m., I placed the 20 mg per liter methamphetamine tubes. The concentration of the drug was increased by 20 mg per liter every 4 days until 100 mg per liter was reached and tested. Also, I switched the water and methamphetamine tube positions every 2 days, starting on day 3, to account for drinking side preferences. Finally, on day 21, I removed the drug and water tubes after the read. The mice were then euthanized and their tail samples were collected for future genotyping purposes. These are the methods generally used in the Phillips lab to measure voluntary methamphetamine intake. And so by testing the final selection generation of methamphetamine binge drinking mice for 2-bottle choice with escalating methamphetamine concentrations, we found that the high and low binge lines methamphetamine consumption significantly increased from each previous concentration. We can also see that the high and low binge methamphetamine intake are significantly different from the control binge, supporting the effect of TAR1 on methamphetamine intake. However, the high and low binge lines did not significantly differ in the methamphetamine intake, except for the 80 mg per liter concentration. And so by comparing the total methamphetamine consumption for my 2-bottle choice data and the Phillips lab 4-bottle choice data, we can see that while the highs and lows both follow an upward trend across concentrations, the binge mice consume dramatically more in the 4-bottle choice paradigm than the 2-bottle choice. More specifically, for concentration 100 mg per liter, the highs under the 2-bottle choice consume about 14 mg per kg on average, while the highs under the 4-bottle choice consume about 43 mg per kg on average. In humans, 13 mg per kg is binge level. Similarly, the lows under the 2-bottle choice consume about 14 mg per kg on average, while the lows under the 4-bottle choice consume about 30 mg per kg on average. Most importantly, in the 2-bottle choice, the high and low binge lines do not differ significantly for every concentration, unlike in the 4-bottle choice. This indicates that the TAR1, M1J, and 1J modifiers of methamphetamine intake in a binge model have a lesser role in modifying the TAR1, M1J, and 1J effect on lower levels of methamphetamine intake. In conclusion, methamphetamine consumption differences between high and low binge under 2-bottle choice conditions are smaller than under the 4-bottle choice conditions. We have also found that high and low binge mice consume significantly higher methamphetamine than control binge mice. Thus, TAR1 impacts binge level methamphetamine intake. Finally, the success of selective breeding for the 4-bottle choice from the TAR1, M1J, and 1J population supports the presence of genetic modifiers that impact binge methamphetamine drinking, but have a lesser role in modifying the TAR1, M1J, and 1J effect on lower levels of methamphetamine intake. As for future directions, the Phillips lab will utilize genetic analyses like those used to find TAR1 to find the genetic modifiers. Funding for this project was provided by NIH, NIDA, Department of Veteran Affairs Merit Review Grant, and the VA Research Career Scientist Program. I would also like to say thank you to everyone at the Phillips Lab, including Dr. Tamara Phillips, Dr. Cheryl Reed, Herway, Jason, Sarah, Sam, John, Dr. Janessa, and Dr. Coulter. I'd also like to say thank you to OHSU, VA, Saturday Academy, ACE, and Lindell. Thank you. Do you have any questions? I have a quick question. So I'm not sure if you mentioned this, but I'm just wondering where that TAR1 receptor expression is localized.
Like, I think you're asking like where exactly it is. I think it's located on chromosome 10.
Oh, awesome. I was curious, like, is it expressed throughout the entire body, or is it like just, I heard that it was expressed in the pancreas. I'm wondering if there are other spots where the receptor is.
I think it's expressed in some other places, but I'm not really sure where exactly.
Okay, thank you.
Thank you. Someone asked what were the findings on the side preference for the mouse. So we changed the two positions so to account for the side preferences in case like the mice like just drink from, because like there were two tubes, one on the left side and one on the right side, and so we switched them so in case like a mice likes to stay more on the right side or drink from the tubes that is on the right side instead of the left. And so by changing, we know for sure what they drank was because they actually liked the liquid and not because they preferred to stay in that place. And the findings for that was that they didn't really have a preference. They just went to drink the liquid they liked the most.
Those are great questions. And that was an awesome presentation. Thank you so much. Does anybody else have any questions? Okay, thank you so much. That was just awesome. We really appreciated it. Great presentation. Thank you. And everybody, it is unfortunately, well, I guess not, that we're getting near the end of the presentations. I always love these. I learned so much, and I just appreciate all the professionalism of all of our interns. But we are almost to the end. We are now going to have, again, I apologize if I mispronounce your name, Jaanya?
Yeah, Jaanya.
Jaanya. Okay, I got to put it all together. Okay, thank you. She is a student at Westview High School, and she's going to be talking about playtime for the little ones, how early access assistive technology is enabling play and mobility for children with disabilities. Her primary mentors were Bethany Sloan and Carrie Philippi, and her mentors organization was Dornbecker Children's Hospital at OHSU. So if you are ready to go, we're ready to listen to your presentation.
Yeah, thank you.
Thank you.
Screen. Hi everyone, my name is Jaanya, and this summer I interned under Dr. Bethany Sloan at OHSU, and I worked in the early access assistive technology clinic, and I learned about and we examined how assistive technology is enabling play and mobility for children with disabilities. So first, this summer, I learned about this clinic, and some of the main goals of this clinic was to encourage play and participation for children with disabilities. It's staffed by occupational therapists, physical therapists, and speech language pathologists with a focus on increasing access to technology for literacy, mobility, play, and communication. This clinic is targeted towards children in the age range 0 to 3 with motor impairments and with limited or no access to assistive technology previously. And one of the other goals of this clinic is to promote use of assistive technology starting at a younger age to help reduce achievement gap between children with and without disabilities. So here's some examples of assistive technology, because before this summer, I was never aware of what assistive technology was or what it looked like. So on the left here is a Go Baby Go car. Go Baby Go is an organization which my mentor actually co-directs. And Go Baby Go makes modified ride-on cars for kids with limited mobility for kids age 0 to 3. In the middle, there's a video of an adapted book, which I actually got to make this summer, which I thought was amazing. And adapted books are also included in assistive equipment. And these are books that are made with kids for kids with disabilities to make them easier to access with visuals popping out and like adding tools to make the pages easier to turn. On the right, on the top right, there's a jelly bean switch, which is a button that you can press in order to navigate small access areas. So for example, you could pair this jelly bean switch with your iPad, and instead of having to navigate a small area, you could press the button, or you could pair it with a toy and press that button instead of having to navigate small access areas. Finally, there's a little step-by-step, which is a communication device where you can record and play back messages, and this helps to enhance and foster communication for kids with disabilities. So one of the main goals, as I stated, for this clinic is to promote AT at a young age, specifically from birth to 3 years old. And this is because it helps to foster mobility and reach developmental milestones in a way that works for children with more complex disabilities. It also helps to decrease the gap between children with and without disabilities, and the younger you start, the better it is. And it's beneficial to start early for children who may be at risk for certain types of disabilities. Some obstacles that prevent widespread usage of assistive technology currently for children between 0 to 3 is that Medicaid and insurances continuously deny power mobility for younger kids. And they also deny being able to switch out or modify devices before a period of 5 years, which is unfortunate because children, especially when in this age range, do grow a lot and they change, which is why it's important to be able to modify your devices. And there's also misconceptions that younger children will not be able to operate the devices. So the pathway to making AT more accessible for younger children is having continued research to show that assistive technology is beneficial for younger children, specifically 0 to 3. And this is kind of where my research project comes in, because I was working on a de-identified patient repository to help support a research paper that is going to be about how the early access assistive technology clinic is structured at OHSU and how it's benefiting kids from 0 to 3. So my main project this summer was creating a de-identified patient repository for this clinic, which included 43 patients from 2018 to 2021. You can see a picture. The Excel sheet was much larger, but here's kind of what the general consensus looked like. So there was 40 different categories to find information for, and the main purpose was to use for research to make AT more accessible. Also, all the data would be uploaded into REDCap, which would then be able to identify trends in therapy, analyze outcomes, and better match children to appropriate protocols. So how is the de-identified patient repository created? So there were basically 3 main steps. So number 1 was data collection from Epic, and Epic is a medical software that OHSU uses, which contains electronic medical records, which I was able to obtain data from of patients in this clinical study. Number 2 was to organize all this data, which was done in Excel. This information was sorted into 40 different categories. Some of them included what diagnosis these children had, how effective was AT, whether they had follow-up treatments. There were many like that. And then the 3rd step was to analyze the repository. So using mass data to analyze and summarize trends. So here is a small screenshot of what some of the summarized data look like. Some of it included what their goals were and where these children currently were with AT use, what the follow-up plans are, whether it happened or not, recommended equipment. Some others were whether they've had exposure to AT in the first place, and really just looking at how this clinic has been structured and how just showing how kids are able to improve with AT, especially in this age group. So we ended up organizing it in a much more clear way, because I feel like that Excel sheet would be kind of hard to read. But me and my mentor, we ended up organizing it into even more bigger categories, such as quality improvement, and then all the data is listed over there, clinical outcomes, and that just includes a few of the categories that were in the repository, as well as demographics of the patients in the clinic, such as age, gender, what location they're from, equipment they've already had before, and many other categories to look for. So the future direction of this research project, as I mentioned, all of the data and trends are going to be uploaded into REDCap, which would perform statistical analysis to better match children to more appropriate treatments and more appropriate equipment. And this information in the repository is going to be used for a research paper that the Early Access Assistive Technology Clinic is planning to publish. My mentor is working on a position paper that talks about the clinic structure at OHSU and share the outcomes on the population served in this clinic. Overall, my experience, I had an amazing experience. I got to learn about assistive technology and this clinic, which I was previously never aware of. I got to assist with clinical research. Along with this, I also got to work on a craniofacial research project, which I thought was amazing. I also got to virtually shadow a few clinics, such as a hemophilia clinic, and I thought it was amazing. I got to see behind the scenes, and it was really amazing to see it. I was also able to learn how to use medical software such as Epic. That's where I mainly collected the data for the repository, and I think it really helped me develop my organizational skills dealing with such mass data. And I can definitely see this as a future career path, because I've always found a healthcare profession interesting. And I think this internship really provided me clinical exposure, which I'm really thankful for. Thank you everyone for listening. I'd like to quickly acknowledge my mentor, Dr. Bethany Sloan, my activity sponsor, Dr. Carrie Philippi, OHSU, Go Baby Go, Saturday Academy, and my teacher monitor, Linda Wolf, for making this internship possible. And thank you everyone for listening.
Thank you so much. That was so interesting. I learned a lot from your presentation. Does anyone have any questions? I guess I was curious about the book that you made. What grade level would that be for? And you mentioned a little bit, but what were some of the features of that book that enabled kids to read it easier?
Yeah, thank you. So that was basically, I think the age or grade level was around first or second grade. And one thing that made it easier was we could add page fluffers on the corners of the books, or like little pom-poms, so it helps make the pages, it makes it easier to turn the pages for kids.
Okay.
Yeah.
It looked great. Did you write the story, or did you take?
Yeah, no, it was already a book, but we helped modify it in a workshop.
Okay. Thanks. Did anybody else have questions? Another awesome presentation. Thank you so much for that. Just really appreciated it. Thank you for sharing. Thank you to everybody who came today, to mentors, co-mentors, to parents, friends, family, everybody that got a chance to come today. So appreciate your support of our interns, and to the interns, once again, great job. Thank you for your hard work this summer. You guys were awesome. Just so much appreciate it. I wish we could do thunderous applause for you in this format. I don't think we can very well, but thank you to everybody that came today. And this is the conclusion of the symposium today. Awesome job. Thank you so much.